left off talking on Monday, looking at early embryonic development in Drosophila. And what I showed you was that the segmentation genes control the homeotic genes. They do that by combinations and concentrations of gene regulators, transcription factors. So that's the specific system for insects, but not even all insects. So many different species, there are variations on a theme. But you have a similar but not identical pathway working, and your homeotic genes are called HOX. And they essentially do the same thing. They give identity. You don't have segments in your body plan, but as I pointed out on Monday, you have segments. If you run your finger up and down your spinal column is segmented. And so that original function that used the Drosophila homeotic genes to segment the body plan and the nervous system is used for you to segment your nervous system. You use a different set of genes to segment and identify anterior versus posterior, dorsal versus ventral. And so we're going to make the switch now to mammals, and specifically humans. The basics are still the same. That's why I want to emphasize that what we're going to pick up now is merely a continuation of what we did on Monday. Even though what we did on Monday was in an insect, and some of the genes are not going to be present in your developmental system, the basics are still the same. We have sets of genes turning on and turning off other genes. And what's really important about the homeotic genes, whether it's the Drosophila homeotic or human homeotic, what's important about them is that they become epigenetic. We've talked about that before. We talked about it in terms of in females, when the Bar-body chromosome is identified in each and every cell, the memory of that, whether you're making the Bar-body out of your mother's X or your father's X, that memory gets incorporated into the chromatin protein structures of the chromosome. But the exact same thing is happening here in flies and in you. When segments are identified early in development, that identity is enforced in the chromatin epigenetically. We talked about that earlier. The definition hasn't changed. It's a modification to the chromosome, basically the histone proteins, and every replication cycle, that modification is reproduced. And so, for example, your nerve genes, the genes that make nerve cell proteins, are in open chromatin in your nervous system. They're in closed chromatin in your blood-forming cells. And so that's what's important coming out of here. Because we're now going to switch to your cells and look in bone marrow. And the system that we're going to look at, the two systems that we're going to look at, is first of all your red blood cells and the protein globin. They form in your bone marrow, and white blood cells, which make antibodies. And so the system which identifies bone precursor cells, is just a continuation from the homeotic genes that identify what different tissues will become. So even though we're skipping ahead in time and we're skipping species, the process is really comparable. And so what we're going to be looking at first is red blood cells and how globin genes are regulated. And then we'll switch to white blood cells and how antibody genes are regulated. Just again, to reinforce this concept, globin genes are open in red blood cells. Antibodies are open in white blood cells, but antibodies are closed in red blood cells. Globin genes are closed in white blood cells. And that open and closure is enforced by epigenetic modification of chromatin. That's what this is all about. That's what we're going to be looking at. Yes?
Does closed mean that basically in white blood and red blood cells, the antibodies are always the same, as opposed to variation in white blood cells?
So what I mean by open and closed, remember we had the different forms of chromatin. So beads on a string, that's open chromatin, as opposed to the 100 angstrom and 300 angstrom fibers, the folded up beads on a string. That's a different form of chromatin, a different structure. That's what we are talking about. That's what open and closed chromatin is. So genes that are going to be used by a cell are typically in open chromatin, and the memory of that is fixed in the histone proteins. Anybody else before we get into this? All right. So we're going to look at red blood cells. Red blood cells are the cells in your body that pick up oxygen in the lungs, circulate throughout the body. The oxygen gradient drops outside the lungs, and so the red blood cells deliver the oxygen to all the cells throughout your body. It does that because the globin proteins are in a structure called hemoglobin. Hemoglobin contains iron, it contains the steroid heme, and it contains globin proteins, 2 alpha type and 2 beta type proteins. And that's what is going to be our focus. Our focus is actually going to be on one part of this system, and that is the beta type globin protein genes. So hemoglobin is a tetramer, 2 alphas, 2 betas, so that's 4 globin proteins. It is a double dimer. So 2 of each, a dimer of alpha type and a dimer of beta type. So that's very simple, very straightforward. That's what's in the red blood cells. So there are alpha type genes and beta type genes. They're very similarly regulated. We're just going to look at the beta type. But that's only part of the problem with mammalian cells. If you think about it, what distinguishes mammals from other vertebrae are the fact that mammals have a placenta. And so as the embryo is developing, the embryo can't get oxygen from its lungs. Very young embryo isn't even going to have lungs yet. So the embryo has to be able to grab oxygen from the mother. And that's a biochemistry problem because the mother, her red blood cells are designed to deliver oxygen to tissues. The embryo is trying to grab oxygen and deliver it to its tissues. So the way that evolution has solved this problem in mammals is that when you were an embryo and when you were a fetus, you used a different set of globin proteins. So the alpha and beta designation, we'll elaborate that in a few minutes, that's the adult set that you all are using right now. But when you were an embryo, when you were a fetus, you had to have a set of globin proteins that was strong enough to grab oxygen from the adult hemoglobin and then deliver that oxygen to the embryonic or fetal tissue. And so that required a very, very different and more elaborate set of globin protein genes. So what we are going to look at is a group of genes grouped together where we have first, we have an embryonic gene, then we have a fetal gene set, and then we have an adult set. And so the development and the regulation of this is that the embryonic gene turns on first, and then it turns off, and then the fetal set of genes turns on, and then they turn off at about the time of birth. All the hormonal changes occurring when women start to have labor will turn off the fetal genes. All those hormones will then activate the adult genes, and there's a handover. So that's the big picture where we're going to go with this. Any questions on the background for what globin is all about?
So, the fetal globin, how long does that stay in effect after birth?
So
those red
there's,
blood cells are still going to be around after birth.
no, and that's absolutely true, but it doesn't matter. So you're right. It's very individual. So it can be days to a few weeks. But then there's a mutation called fetal persistence of fetal hemoglobin, where it never turns off. And those people are perfectly normal. You never find out that an individual has that until they have blood work done. So it's not the end of the world to have the fetal hemoglobins left on accidentally or on purpose.
So they'll transfer O2 just fine
Yep.
throughout? Okay.
Yep, and they'll pick it up better and transfer it just fine. Anybody else on the background? Yeah.
So is there an evolutionary advantage to switching to adult hemoglobin after birth?
That's an interesting question. I have no idea. I don't know. I've never thought about it before. Because the fetal persistence syndrome is absolutely without phenotype. Anybody else? All right. So the alpha type genes and beta type genes each are located in one cluster each on different chromosomes. And so we're going to focus on the beta type, where we have beta and delta genes. So you actually have two different adult type globin genes. They are virtually identical. There's only 1 or 2 amino acid difference. They were formed by gene duplication and then one mutated slightly. They are almost identical. So each one of these boxes is an individual transcription unit with a promoter and a terminator. I'll give you the gene structure, the intron-exon structure, in a few minutes. The major regulator is at the 3' end, and so this is an enhancer. So the regulator for the globin genes is not located at the 5' end of the gene. The regulator is located at the 3' end. That really doesn't matter because most of your regulators are located a long distance away. They all act by the DNA looping over, almost like a pretzel, to contact the promoter. So enhancers literally can be anywhere, even inside a gene in an intron. It doesn't matter. In these genes, they just happen to be at the 3' end. So this is your adult set. Here we have gamma 1 and gamma 2, again, independent transcription units, enhancer at the 3' end of the gene. And then out here, we have epsilon. I think I told you the first day of class, you become fairly well versed in Greek. And so these are all Greek letters. This is fetal and embryonic, promoter, terminator, and enhancer. And so those are your active genes all in one cluster. There's another gene in there, which is a pseudogene. And that Greek letter is psi, for pseudogene. It's located there. So you really have hundreds to thousands of dead genes littering your chromosomes all over. A pseudogene, classically, is a gene whose gene structure suggests that it cannot function. That's not entirely true anymore, since we now know that almost everything is transcribed, and lots of pseudogenes have functions. They just don't have the same original function. And so there's a pseudogene there. It may not be doing anything in this cluster, but that's not true for pseudogenes in general. So here we have the time schedule from early embryonic. We turn on epsilon, then we turn it off, we turn on the gamma genes, then turn them off and turn on delta beta. Beta is your major, delta is your minor. If you have blood work done, you will have alpha beta as your major hemoglobin and alpha delta as your minor hemoglobin. Excuse me, any questions on that so far? And then out here, we have the globin locus control region. So the globin locus control region is a ginormous, huge, multiple enhancer. It's very, very large, and it regulates this whole set of genes. So we have two different levels of control for your globin genes. One level of control is at the individual gene. So each individual gene has its own individual regulator. The globin locus control is like the master regulator for the whole region. So again, the big picture, the way this works, is that what we will do, what I will show you, is that very early in embryonic development, you're going to want to turn on the embryonic genes. But the way we do that is to open the entire region, and the globin locus control will pick out the embryonic gene to turn on. And so that's the big picture for how we're going to start. Now, this is the time sequence, and so we're going to turn on embryonic first, then fetal, and now you are running your adult genes. These are all turned off. There's one more wrinkle to this, and that is, not only is there time course regulation, but there's tissue regulation. So this is done in bone marrow. This is done in the fetal liver. This is done in the embryonic, this is done in the embryonic yolk sac. And so not only do we have regulation over time course, but every time we change the use of the genes, we also do it in a different tissue. Any questions on this so far? All right, so again, I want to emphasize one thing on that top board. 1 second. That's the paradigm. That's what's all important, is that development is constantly changing. As we go from early embryos to late embryos, as we form anterior, posterior, dorsal, ventral, tissues come on, homeotic genes say that this is going to be a thorax, this is going to be a head, and then within the head, other genes turn on and say, well, this part will be an eye, this part will be a nose. So that's a constantly changing set of regulatory events. And so what that means is it's all run. Change means that the concentration of individual regulators and the combination, so some are going to turn off with time, and as time marches on, just like we saw in the early embryos, we went from the maternal genes, the bicoid, the nanos. We went from the maternal genes to the segmentation genes. I didn't give you their names. They're in your textbook. Those got turned off, and we went to the homeotic genes. And so that progression is just going to continue right through adulthood. And so as we start switching over from homeotic genes, in the yolk sac, we turn on for the concentration and combination of gene regulators is perfect in the yolk sac to open this up. And what happens is that the globin locus control concentration and combination of transcription factors becomes perfect in the yolk sac to activate the globin locus control region. And so in the yolk sac, this whole region opens up. The entire region. But the combination and concentration is perfect for the GLC and for that enhancer, which then turns on that gene. And so in the embryo, in the yolk sac, you begin making the epsilon gene And the same thing is happening on a different chromosome. We're making one of the alpha-type genes. And so we're making the first of the hemoglobin proteins. So now the embryo is competent to grab oxygen at that blood barrier in the placenta to begin feeding oxygen on its own to tissues. Everybody see how this works? The whole region is going to open up. Any questions on that? Yeah.
Go ahead. Just to clarify, the GCL is what starts each enhancer at a particular time.
Right, so that's the master regulator for the entire region, but it knows which gene to hit.
At each time.
At each time point, in each tissue, it knows in the yolk sac to turn on the epsilon gene.
Thank you.
You have a question?
Yes, I just wondered, when that's the open, when that's towards the bottom line?
Concentration and combination, over and over and over again. Concentration and combinations of transcription factors that are always changing as development progresses. Individual regulatory genes will turn off, other regulatory genes will turn on as time progresses. Any questions on that?
So, GLC turns on the yolk and we receive oxygen.
Anybody else? So, as time marches on, the concentration and combination of transcription factors will change until in the yolk sac, it's no longer the proper combination and concentration to run this system, and this will close, and instead, with time, the combination and concentration in the yolk sac changes, it turns off there, and it turns on in the, we now are further developed, we have a fetal liver. In the fetal liver, the combination and concentration becomes correct. And now we open up the whole region again. The whole region opens up, but the GLC is now recognizing these two enhancers because the combination and concentration of transcription factors that bind to these are the correct ones. It doesn't recognize those enhancers. It doesn't recognize that enhancer. And so now the entire region is open, but the gamma genes are specifically turned on. Time marches on. As time marches on, we are going to close down the system in the liver, and then shortly before birth, the combination and concentration of gene regulators in the bone marrow cells becomes appropriate. The GLC shuts down in the liver and opens up in the bone marrow. And now that is what you are running on now. And so you will use this system forever. Any questions on how this works? So each one of these genes genes are identical in structure. All the genes, alpha and the beta genes, came out of a single gene that duplicated and diverged. And so that's going to be a common theme towards the end of the course when we hit the chapter on evolution. Gene duplication and then random mutation, selection for good function, is what allowed the development of placental development. There could not be placental development unless there was this multiplicity of globins with different oxygen binding ability. Any questions on how this system works? So now, we are going to focus on the second part that's coming out of the bone marrow. So there are dozens, perhaps hundreds of different white blood cells, all having a common origin. We are going to focus on one specific type called B cells that make antibodies. So the white blood cells and red blood cells have a common origin in terms of the tissue that gave rise to them. Antibodies are the proteins that you make. They have specificity or antigens. That's the other part. So antibodies' function is to recognize a structure, sort of like a hormone receptor. Now when antibodies recognize a structure, they bind to it, and if that structure is a bacteria or a virus, it leads to its destruction. If it's a pollen grain, it leads to your allergies. So it's the same system working the same way. In a sense, antibodies are kind of similar to the globin in that we're talking about a tetramer of 4 proteins, a double dimer, 2 of this and 2 of that. So the general antibody structure looks like this. We have 2 light chains designated L. Those are the small proteins. 2 heavy chains designated H. Those are the large proteins. My proportions aren't exactly correct. The heavy chain is 4 times the size of the light chain. So we have a double dimer, 2 L's and 2 H's. Unlike the globin, where there is no covalent attachment, these are all covalently joined by disulfide bonds. And so that's what the SS is. Those are cysteine amino acid residues. They're 2 sulfur-containing amino acids. Cysteines form covalent bonds fairly readily. And so the antibodies are a tetramer, a double dimer, held together by disulfide covalent bonding. Not true for the globin, they're held by hydrogen bonds. Now, what's interesting about antibodies is the fact that you have about 20,000 plus genes. Yet you can make tens of millions of different antibody proteins. And so going back to the 1950s, 1960s, that was one of the great mysteries, is how an organism with a known limited number of genes can make a virtually unlimited repertoire of antibodies. Someone could sit down in a laboratory and design an organic structure, inject it into you, or more commonly, a goat or a rabbit, and within a week to 10 days, that structure will cause the B cells to start producing an antibody which will lock and key, lock onto that structure for its ultimate destruction. So again, it was known way back in the 1950s that you can make an almost unlimited number, millions and millions of antibody proteins from a limited number of genes. And so that was very mysterious until people finally entered the cloning era and began to see the structure of the antibody genes. The mystery was solved. And so part of that mystery has to do with the structure of the protein itself. We have amino and carboxy end of the protein. If you know this terminology, we've had it before. The protein has an amino end and a carboxy end. The red line through there is a line that people identified, I guess, late 60s, as they began to sequence the proteins and were able to get clonally pure antibodies. You go into your body, you can't get clonally pure antibodies unless you have leukemia. And so leukemia is a white blood cell cancer, soft tissue cancer, where one antibody-producing cell begins to expand uncontrollably. And so, excuse me, because one cell is clonally expanded, people were able to get from leukemia patients, each leukemia patient made a completely different antibody. And so as people began to sequence the antibodies from leukemia patients, they discovered something very interesting, that they could literally draw a line through the antibody protein, the tetramer, which cuts the light chain about in half and the heavy chain about in quarters, and identify a variable portion and a constant portion of the protein. So that was the first something unusual. As you look at hundreds of different antibodies from different leukemia patients, what you saw was they all had, almost all had, the same or very, very similar constant part of the gene. The back half of the light chain, the back 3/4 of the heavy chain, but every single antibody had a different amino path, again, on the light chain. And so that was the first clue as to how you can make millions and millions, unlimited number of different antibody proteins. Any questions so far? I've been going on and on. Okay, so that was in, I guess the late 1960s. Then the next big surprise as people began to try to map where the antibody proteins originated from, map the genes, they discovered something which at the time was just mind-boggling, that the variable portion of the gene was coded for separately, genetically unlinked, separately from the constant portion. And for a while, it looked like two genes coded for one protein, which was just heresy at the time. So we have a separate coding part of the chromosome for the variable part of the protein, separate from the constant part of the protein. And so that's where we're going to pick up the story. As people began to clone and sequence the antibody genes, the immunoglobulin genes. Let me put that word up on the board. So this is immunoglobulin as opposed to globin. Please do not mix up those two words. If I ask for one and you give me the other, I won't give you any points. Make sure you get that terminology correct.
When you're saying that you said that they code for the same thing, are you saying they code for the exact same thing or different parts?
So the constant portion, what we see is the exact same constant portion connected to hundreds, thousands, millions of different variable portions in the protein.
But no, yeah. When you said the thing, it was heresy. That two things code for the same.
So the heresy is two genes coding for one protein.
Okay.
So the dogma back then was one gene codes for one protein. But now we seemingly have a situation because the variable coding part was genetically unlinked from the constant coding part. It looked for a while like two genes coded for one protein. Anybody else? Okay, so the answer came in looking at the DNA sequence of the antibody region. And so we are going to look, let me label some of these. So we have kappa or lambda. There are only two different light chains. We are going to look at kappa. For the heavy chains, there's alpha, gamma, M, let's see, and a couple of others. The heavy chain gives its name as IgA, IgM, I think it's IgG that people with asthma have a problem with. So the name of the antibody comes from different heavy chains, an A chain, an M chain, a G chain, connected to either kappa or lambda. We'll go through the DNA first, and then we'll review the biology of how this system makes antibodies. So again, any general questions on the terminology before I show you how two different regions, genetically unlinked, can make a single protein? So two different regions, genetically unlinked, but on the same chromosome. So the kappa genes are all located on one chromosome. The lambda genes are all located on a different chromosome. And the heavy chain genes are all located on a third chromosome. Each one is done independently. We'll look at the biology at the end. And so what we have is a set of variable region I like have to call them cassettes. They're really not genes. They are part genes. I'll explain that as we finish up the drawing. Cassette is really a good word to describe what's going on. So we have the body of the variable portion, the front portion. Remember now, these are the kappa genes. The variable portion comes in 2 exons, L1, V1. The space in between is an intron. And we have L2, V2, L3, V3, L200, V200. Each one of those V regions, and so the whole thing is called the V region, but the V region is in 2 exons with 1 small intron. Each one has its own separate promoter. No regulator, that's really important, no terminator. So it's not a transcription unit. The transcription unit has a promoter, regulator, and terminator. All we have here is a promoter. And then out here, we have the kappa constant region. It's got a transcription terminator, and here, takes me a second to draw all these, we have joining regions, J1, J2, J3, J4, J5, and J6. The joining regions are little mini exons. And then, in the intron in between J6 and kappa constant, we have the regulator, the gene regulator, genetically unlinked to the promoter. So the way this stands, we have 2 separate gene regions on the same chromosome. We have a set of cassettes with promoters that can't be used because there's no regulator to turn them on. And we have the regulator and the other half of the gene and the transcription terminator half a chromosome away. Not exactly half, but long ways away. Everybody see? This is the paradigm for all 3 genes. Remember, there are 3 different ones. There's lambda and kappa light chain, and then we have all the heavy chain. They all work about the same way. Not exactly, but similarly. So we have a problem. The problem is to make an antibody protein, we have to take a variable portion and connect it to the constant portion. The variable portion will bring its promoter with it. The constant portion has the enhancer and the terminator. Any questions on this structure?
So the V's don't have promoters or terminators. The constant region only has a terminator, no promoter, just an enhancer.
Right. This is called the germline gene sequence. So this is the sequence, this is human. This is the sequence of your kappa light chain antibody genes